Okay, Assalamualaikum warahmatullahi ta'ala wabarakatuh. This morning, screen recording will cover the rest of my slide notes. Okay, the YouTube video that I shared before, I think already cover most of the physiological thermal strategy. Okay, today I'm going to cover this one, biochemical thermal strategy. Okay, and also additional notes from the second notes that I attached for you guys last Monday. All right. Okay, biochemical thermal strategy. Before we go to this, a bit refreshment. In all living things, we have these macromolecules. Okay. In our body, we have protein, we have lipid, we have nucleic acid, we have carbohydrates. In between these macromolecules, we have bonds, okay, interaction and bonds within and between the macromolecules. So the bonds, hydrogen bond, hydrophobic interaction, van der Waals forces. The chemical bonds can be disrupted by high temperature. Okay, and different bonds, okay, will have different temperature that can break these bonds here. Okay, as you can see here, hydrogen bond, van der Waals bond, disrupted by high temperature. Okay, hydrophobic interaction here, stable at high temperature, but low temperature might break or might alter this hydrophobic interaction. Okay, now relate that bonds with this term here, membrane fluidity. Okay, van der Waals force hold membrane lipids together. Interaction between phospholipids are strong, but membrane must remain fluid enough. Okay, we have phospholipid bilayer at membrane, right? So this phospholipid bilayer membrane function is selectively allow molecules or particles to cross them. Okay, so any changes in the phospholipid structure itself or any changes in membrane itself, okay, membrane fluidity can affect any movement here, protein movement. Okay, low temperature cause membrane lipids to solidify, so the membrane become more rigid, so it will hinder or impair the protein movement. Okay, high temperature will cause the membrane become fluidity, so more liquefy. Okay, so when it's too high temperature, it can cause defective or low effectiveness as permeability barrier. So when the membrane cell exposed to very high temperature, so the membrane becomes so liquefied or tend to be liquefied, so that membrane will allow any particles to cross them. So in terms of transportation of particles, it becomes so easy. So but in terms of we can say safety of the cell, so inside cell can expose to many harmful particles or okay. This membrane fluidity relate with protein function and phospholipid fluidity. Okay, so it relate with the structure of the phospholipid bilayer, whether saturated fatty acid change or unsaturated fatty acid change, okay, double bond, the presence of double bond, or protein function, any protein molecules that embedded in between the phospholipid bilayer. Okay, so how to maintain the membrane fluidity? By remodel the membrane, okay, which we call homeoviscous adaptation. So this remodeling will alter the fluidity of the membrane. Okay, homeoviscous adaptation preserve membrane fluidity across different temperature by changing the membrane composition. Okay, so regulating membrane fluidity is especially important for this type of animal, poikilothermic organism. Okay, yeah, because they tend to tolerate with the environmental or ambient temperature. So what happened in membrane remodeling mechanism? So there are two materials or two compositions that can be altered. Okay, one is the phospholipid itself, and another one is the cholesterol content. So phospholipid bilayer, the membrane have all phospholipid bilayer sit together. Okay, side by side. Okay, and this layer, outer layer, and also the inner layer. Remember your cell biology. Okay, and the cell membrane, in between the cell membrane, the phospholipid bilayer, we have cholesterol structure embedded in between. Okay, so we look into this phospholipid composition here. In changing the phospholipid composition, we the system, the cells, okay, the body system, the body cells of that organism can change in terms of fatty acid change length. Okay, so whether it's longer or it's shorter. Okay, when the fatty acid change length is short or become shorter, so the cell membrane will become more fluidity. When the fatty acid change longer, the cell membrane become more solidified or more rigid. Okay, so in terms of phospholipid saturation, okay, this one relate with the double bond that present at the fatty acid change. Okay, phospholipid bilayer, this fatty acid change over here. Okay, when that fatty acid change, fatty acid change have double bond inside and in their change. So the feed of this one, okay, this fatty acid as fatty acid change will have kings. Okay, so a lot of double bond will make this phospholipid acid fatty acid change have kings. When there's a lot of kings, these kings, okay, it makes the membrane cell become more fluidity. When we compare this lipid layer, okay, we make it bilayer. So as as we compare here, all of this fatty acid is straight, okay, due to less or no double bond at all. So saturation of the fatty acid change is higher. This one, a lot of double bond, okay. This makes the fatty acid change have kinks, a lot of kinks, so the cell membrane become less rigid, so more fluidity, okay. So the protein movement or the particle move easily in this type of cell membrane. Okay. Next, composition of phospholipid that we can alter is the phospholipid classes. There are two types of phospholipid. Okay, one is phosphatidyl chlorine and another one is phosphatidyl ethanol anide. Okay, in warm acclimated cells have these type of phospholipid classes. So in cold acclimated cell have this type phospholipid classes. So this one you can imagine that, or you can relate that phosphatidyl chlorine class is more makes the cell member become more fluid, okay, warm acclimated. Become more rigid, because in cell in high temperature tends to become more liquefied. So when we change the cell membrane of these cells with phosphatidyl chlorine type of phospholipid, that cell membrane will become more solidified in high temperature. Okay, so vice versa for for the second one here. This cells is in cool environment at all times, so the cells tend to become rigid or solidify. So the cell will have or change the composition of the phospholipid classes with this type here. It will make the cell membrane become more liquefied in cold environment. Okay. Okay, that basically what happened at membrane remodeling approach.